Thank you. I would like first to thank the organizing committee, all the people involved in the organization of this great meeting, and especially Josiane for inviting me to attend this wonderful meeting, and yesterday night a wonderful gala dinner. My talk is focused on the role of protein phosphatases, and particularly PP2A, on redox signaling in acute inflammation and in acute pancreatitis as a model of acute inflammation. This one. Okay. Acute pancreatitis starts with abdominal pain. Many patients, they feel like stomach ache and they think that it's a normal ache. They wait for the disappearance of this pain, and then it's too late. When the pain still keeps for a long time, they go to the hospital, and when they enter in the hospital, they start to develop failure at the level of the lungs. They develop dyspnea, hypoxemia. They may develop kidney failure, oliguria, and azotemia. And then the disease, which initially is local disease, local inflammation, it starts to develop to become a severe systemic disease. The causes, major causes of acute pancreatitis are first alcoholism, mainly in males, and gallstones, mainly in females. Acute pancreatitis shouldn't be considered just a local disease, because the local inflammation of the pancreas triggers the release of pro-inflammatory cytokines, which goes to the blood, and then there is an extrapancreatic affectation, mainly in the kidney and in the lung. So we have acute tubular necrosis and respiratory distress with damage in the alveolar wall in the lung. And there is, in those cases of severe acute pancreatitis, is death by multiple organ failure. And this, the percentage of death is quite important, because it's around between 20 and 30% of cases of severe acute pancreatitis. At the end, the severe form of the disease is like a septic shock. So in some way there is a great relationship between acute pancreatitis, the severe form, and those severe forms of systemic inflammation, systemic inflammatory response. We have developed in our lab experimental models of acute pancreatitis with the mild form, which is induced by cerulein, an analog of cholecystokinin, which induces hyperstimulation. And we also developed a model of necrotizing pancreatitis as a severe model of the disease, induced by infusion of taurocolate into the biliopancreatic duct. And we also have in vitro models to study redox signaling and to silence genes involved in the cascade. We have here the complex inflammatory cascade, which is supposed to be triggered by two hits. First damage, which is amplified by second hit due to either hyperstimulation with cerulein or with bile acids, which not only damage the membrane, but also they trigger a cascade in which oxidative damage products, DMPs, are involved, especially oxidized lipids. And a major part of the cascade is through MAP kinases, NF-kappa B, and CREB and CBP, which is involved in the acetylation of the chromatin. Regarding the MAP kinase cascade, we have upstream MAP kinases the MAP kinases, which phosphorylate MAP kinases. And when we studied this MAP kinase pathway in acute pancreatitis, we found a clear increase in all the 3 major MAP kinases. And we found only one MAP kinase which was upregulated, which was activated in the course of pancreatitis. And this could explain, at least in part, the activation of MAP kinases. But we should consider that apart from the upstream kinases, which activate MAP kinases, we also have phosphatases, which normally they dephosphorylate MAP kinases and they keep them in the inactive state, as we saw yesterday. And among protein phosphatases, we have 2 types of protein phosphatases which are redox sensitive. We have serine threonine phosphatases, and among them PP2A, which has in the catalytic subunit thiol groups, redox sensitive. And among tyrosine phosphatases, we have all of them, most of them, they have thiol groups which are also redox sensitive, so they could be inactivated when there is oxidative stress. When we looked at the redox status in the pancreas during the inflammatory cascade, we confirmed that there was glutathione depletion, which was already reported, and this is typical for acute inflammation. And our surprise was that this glutathione depletion was not associated with an increase in oxidized glutathione. So there is no significant change in the glutathione redox status. And when we looked at cysteine and cysteine, we found an increase in both. We didn't find any change, significant change, in the level of protein glutathionylation. And it was very remarkable that we found a very marked increase in protein cysteinylation in the course of acute pancreatitis. At the basal state, it was undetectable. And in the course of the disease, there was an important increase in protein cysteinylation without glutathione oxidation. We confirmed this thiol oxidation using different switches with fluorescein, labeling the oxidized thiols, or using monobromo bimane. And in both cases, we found an increase in the oxidized form of the thiols. So we propose this type of oxidative stress as a disulfide stress, which is associated with glutathione depletion, with glutathione hydrolysis, which releases cysteine and gamma-glutamyl cysteine, and we detected the protein cysteinylation, and we also detected protein gamma-glutamyl cysteinylation. So we studied further which were the targets of these mixed disulfides. And we found different types, two major types of targets. Ones which we call redox buffers, such as albumin and the inhibitor of ribonuclease. We detected that by Western blotting, comparing the reducing conditions and the non-reducing conditions. So with the inhibitor of ribonuclease, we found a decrease in the signal in the Western blotting. And we also detected redox-sensitive thiols in protein phosphatases, in PP2A, and in tyrosine phosphatases, particularly in SHP1 and SHP2. This would be redox signaling thiols. We confirmed the oxidation of thiols in PP2A. We performed diagonal electrophoresis, and when we performed the Western blotting, first in the diagonal electrophoresis, we performed first a non-reducing electrophoresis, and the second dimension is with reducing conditions. And when we detect a signal, a dot, above the diagonal, then it means that there is due to intramolecular disulfides. If the dot is below the diagonal, it indicates that the disulfide is intermolecular. So in the case of PP2A, our results suggest that there are intramolecular disulfides which are formed in the course of pancreatitis. We confirm that these thiols are redox sensitive because by giving N-acetylcysteine to the rats subjected to acute pancreatitis, we prevented the loss of activity of PP2A and of tyrosine phosphatase activity, which was found in the course of acute pancreatitis. And we performed experiments in vitro to confirm the key role of PP2A in the inflammatory cascade. When we silenced PP2A, the catalytic subunit, and we found that just a minor, a slight decrease in the expression of the subunit triggered a huge increase in the phosphorylation of ERK and P38 alpha. So this protein phosphatase is critical to amplify the inflammatory cascade, which is mediated mainly by ERK, activation of ERK and P38. And we also detected an increase in the levels of pro-inflammatory cytokines, TNF-alpha, IL-6, and IL-8 in these cells. There were 266 cells, which are mouse cells from the acinar pancreas. So our model suggests that under physiological basal conditions, we have active protein phosphatase PP2A, which is blocking the cascade of the MAP kinases, whereas in acute inflammation, particularly in acute pancreatitis, there is inactivation of the protein due to disulfide formation, and then the brake is released, so there is an activation of the pathway of MAP kinases, which is the possible role that has PP2A in chromatin remodeling, because there are histone deacetylases which are sensitive to phosphorylation. They are, when they are phosphorylated, they suffer translocation to the cytosol, and when they are unphosphorylated, they remain in the nucleus. So we studied the remodeling of the chromatin. Chromatin can be in a compact state, in a silent state, and in order to induce the inflammatory cascade, it needs to be open, and mainly it's open through acetylation of histones. We suggest that in inflammation we have an initial phase of activation, which is due to recruitment of histone acetyltransferases mediated by the activation of MAP kinases and inactivation of protein phosphatases. And in the resolution of inflammation, there is recruitment of histone deacetylases and a recovery of the activity of protein phosphatases. In histone acetylation, a major part which causes the opening of the chromatin is the charges of the acetyl group, because acetylation is localized in lysines. Lysines have a positive charge which favors binding to DNA, and it causes the compactation of chromatin. Whereas when there is acetylation, then this negative charge favors the opening of the chromatin and then the accessibility of the transcription factors to trigger the inflammatory cascade. We perform experiments by chromatin immunoprecipitation to detect which transcription factors and which histone acetyltransferases and histone deacetylases were bound to the promoters of pro-inflammatory genes. We did that with specific antibodies and PCR of the promoter regions of these pro-inflammatory genes, and we found recruitment of histone acetyltransferases, mainly CBP, to the promoters of different pro-inflammatory genes. So the model we propose is that in which, under basal conditions, we have no recruitment of histone acetyltransferases, whereas in the active form, either for early genes, we identify two types of induction of genes involved in the inflammatory cascade. Ones which are they are induced very quickly, such as ERG1, the transcription factor, and others which are the typical ones, late genes associated with the inflammatory cascade. And in both cases, we have recruitment of histone acetyltransferases, such as CBP and PCAF, mainly CBP, in both cases. And associated with this recruitment, we have also typical recruitment of NF-kappa B. We have here the similar model. We did it in different times, confirming the recruitment of the histone acetyltransferases. This is for TNF-alpha promoter, and with that also for INOS promoter, with a similar pattern, recruitment also of CBP in the course of the induction. And in the basal state, we found recruitment of histone deacetylases. In order to confirm this model and the role of PP2A in the remodulation of the chromatin in vivo, we studied one drug, which is pentoxifylline. This is a derivative of the methylxanthine, and which it blocks the production of TNF-alpha, but the mechanism of action of this drug was not elucidated, and we, in fact, this was the starting point, the origin of all our study, because we found that pentoxifylline, in fact, is acting mainly through PP2A, because the decrease in the activity of PP2A in acute pancreatitis was prevented by pentoxifylline without affecting other protein phosphatases, such as PP2B and PP2C. And the recovery of this activity by pentoxifylline was, at least in part, mediated by the recovery of the glutathione levels. There is glutathione depletion, as I said before, and pentoxifylline was able to recover the levels of the major antioxidant, which seems to prevent thiol oxidation in the protein phosphatase. And when we studied chromatin remodulation, we found that the recruitment of histone acetyltransferases, which occurred in the course of pancreatitis, was prevented by pentoxifylline in the different pro-inflammatory genes. We also confirmed that the prevention in the recruitment of histone acetyltransferases was associated with prevention of recruitment of transcription factors, mainly NF-kappa B, with pentoxifylline. So our model that we propose as a mechanism critically involved in the induction of the inflammatory cascade, is mediated by disulfide stress as a subtype of oxidative stress, which is associated with glutathione depletion, with glutathione hydrolysis, but without glutathione oxidation. Disulfide stress is mainly associated with protein cysteinylation, and it would cause the inactivation of protein phosphatases, mainly PP2A. This inactivation favors the opening of the chromatin, the recruitment of transcription factors mediated by the phosphorylation through the MAP kinase cascade, and then the induction of the pro-inflammatory genes in acute inflammation, particularly in acute pancreatitis as a model of acute inflammation. And I would like to thank all the members of my group, especially Mariluz Moreno, Anabel Gil, and Javier Escobar, who did most of the work, and the collaboration with Antonio Martinez Ruiz in Madrid, who helped a lot in the design of the experiment, to detect mixed disulfides, and also my long-lasting collaboration with clinical friends specialized in acute pancreatitis, Luis Sabater and Luisa Parisi, with whom I have many talks in the countryside in Spain, enjoying life and science. Thank you very much for your attention.
Thank you very much. This talk is open for questions.
Very interesting talk, very interesting data. You attribute the loss of glutathione to increased degradation. Is it possible that you can also have some increased efflux of glutathione, either by MRP proteins of oxidized glutathione or release of reduced glutathione, as was observed for cells close to apoptosis?
We measured reduced and oxidized glutathione in the blood and in the plasma, and there was no increase.
Interesting too. What's the effect, if any, of pentoxiflin on the protein tyrosine phosphatases in general, and the SHP2 and SHP1, which you have showed decreased expression?
We're studying now the effect on tyrosine phosphatases. The effect of pentoxiflin is a little bit complex. I didn't have time to show it. This is an inhibitor of phosphodiesterase, and it prevents the decrease in cyclic AMP levels, which also affects PP2A activity. We have found that in vitro, there is a very rapid decrease in PP2A activity associated with a loss of cyclic AMP levels. And later on, there is inactivation of the protein. So the situation is a little bit more complex, but the inactivation mediated by thiol oxidation is very important to keep inactivated the enzyme. So in the long term, especially in vivo, it's very important the long-lasting inactivation of the enzyme due to oxidative stress. And the role of pentoxiflin, since it prevents glutathione depletion, it should affect also tyrosine phosphatases. Yeah. So it's not only mediated by PP2A.
Yeah. And the other thing is chronic inflammation has been shown to increase the expression of phosphatase like SHP2, PTP1B in the pancreas. So here you see decreased expression. Is it more because it's acute versus chronic inflammation, which is affecting the different effects on expression?
Well, these genes, especially tyrosine phosphatases, they are induced in the long term when there is oxidative stress. I imagine to try to adapt to this condition. In any case, I should say that we have to we should perform systems biology, and we should take in mind that we should have an overall picture of the story, because this is the starting point of the inflammatory cascade. If we wait some hours later, 24 hours, then the activation of MAP kinases, especially ERK, it mediates an adaptation response, because the phosphorylation of ERK phosphorylates MYC and upregulates gamma-glutamyl, their synthesis, gamma-glutamyl cysteine ligase. So it promotes glutathione synthesis in the long term, later on. So it's very important to block the inflammatory cascade at the beginning. Later on, there are adaptive mechanisms mediated by phosphorylation which are beneficial.